So thank you very much, Emma, for the kind introduction. Thank you very much also, Dr. Pavlovic, for organizing this such a nice webinar series, okay? So today I'm going to talk to you about this project that we have in the lab where we are performing an integrative analysis of cardiac epigenetics. Okay, so as probably everybody knows in this webinar, during the development of hypertrophy and heart failure, there is a change in gene expression that is controlled by different epigenetic factors like DNA methylation, histone marks, or transcription factors. So when we started this project, we hypothesized that chromatin structure may be another factor that is involved in this process, okay? And we hypothesized that because it's very well known that chromatin can be organized to conform active or inactive environments where the genes that are located in these regions tend to be transcriptionally active or transcriptionally repressed. So when we started this project, we then decided to use two different models of heart failure. The first one was transverse aortic constriction, okay? So this one is a pressure overload model, very well known in the field. We're just performing a small knot in the aorta of the animal. We are gonna be able to induce hypertrophy of heart failure if we wait long enough. The second model that we decided to use for this project was a cardiac-specific knockout mouse for CTCF. Okay, so CTCF is a protein that is ubiquitously expressed in the nucleus of the eukaryotes that is able to bind along the entire genome by using different combination of its 11 zinc fingers. So the main function of this protein is to mediate the genome organization. So bringing together regions that are far from each other in the same chromosome or in different chromosomes, okay? So the thing is that CTCF is synonym of chromatin structure. So when you decide to start working in chromatin topology, CTCF is gonna become one of your main focuses, okay? That's why we started working with this protein. So the first thing then that I did when I started working on this project was to take advantage of this experiment called hybrid mouse diversity panel. So this experiment was performed by two of our collaborators here at UCLA, Dr. Yibing Wong and Dr. Jake Lucis. And what they did in this experiment was to induce heart failure in 83 different mouse strains using isoproterenol, okay? So after that, they analyzed changes in gene expression and in the phenotype of these animals. So when I got access to this data, the first thing I did was to check what was happening with CTCF levels in all these mouse strains when you induce heart failure. And then, as you can see here, in the majority of these mouse strains, the CTCF level decreases after inducing heart failure. So this one was confirmed using an RBM, neonatal rat ventricular myocytes, where we induce the disease phenotype using isoproterenol, the same drug that they use for the HMDP panel, and also using a different agonist called phenylephrine. So in this case, you can see again that when we treat these NRBMs with these two agonists, we have less CTCF. When we look for a correlation between CTCF level and heart size, what we found is that there is a negative correlation between CTCF expression and heart size, okay? So that's something that you can see here in this panel. So here, each of these dots represent one of the mouse strains that I mentioned before. In the x-axis, we have CTCF level, and in the y-axis, we have three different traits that are related to heart size. So here we have total heart weight, right ventricular mass, and left ventricular mass. So you can see here then how the lower is the amount of CTCF in the heart, the bigger is the heart, okay? So these two pieces of evidence suggest or show that when we induce heart failure, we have less CTCF. But what happened in the opposite direction? What happened with the heart when we deplete CTCF? So to answer this question, we generate our own inducible cardiac-specific knockout, CTCF knockout animal, okay? To do that, we use the Merck-Rimer system. And as you can see here, using this system, we are able to deplete up to 80% of the CTCF protein that are expressed in the cardiomyocytes. So when we study the phenotype of these animals, what we found is that compared to the controls, the CTCF knockout animals develop a very strong disease phenotype that is characterized by dilation of the left ventricle in both diastole and systole, and also characterized by a dramatic reduction in the ejection fraction. Okay? Here I'm showing you two, this gray violin is also representing the TAC animals. Okay, so here again, when we perform TAC, we see an increase in the size of the left ventricle and a decrease in ejection fraction. But these differences are more dramatic when we deplete CTCF. So this echo data then suggests that the knockout, the heart of the knockout animals is bigger and is not working very well. So to confirm that, the first thing that we did was to perform histology of this heart. And as you can see here, it's true, after depleting CTCF, the heart of this animal increases. So the heart of the CTCF knockout animal is much more bigger than the control, and the size of these hearts are similar to the one that we have when we perform TAC. To also study cardiac activity, we took advantage of the M mode of the echocardiograms. So here you can see how the heart of the knockout animal is barely beating compared to the controls. Okay? So this experiment then showed that the inducible inactivation of CTCF causes heart failure. To confirm that alteration of CTCF level correlates with heart failure, we also study CTCF levels before and after the implantation of the LVAD, a left ventricular assistant device in patients. Okay, so the heart of this patient is a hypertrophic heart, but the ejection fraction of this heart is coming down so fast that they need an extra help, an external help, to pump the blood out of the heart. That's why they undergo the implantation of this device. Okay, so when we study CTCF levels, when we study the difference in CTCF expression before and after the implantation of the device, what we found is that after the mechanical unloading of these hearts, we see an increase of CTCF levels. Okay, suggesting or showing that CTCF, the amount of CTCF in the heart respond to heart failure, but also to the treatment of the failure of the disease. So then the summary for this, in this first part, we can say then that heart failure promotes down regulation of CTCF, that CTCF levels negatively correlate with heart size. I also showed you that CTCF depletion induces heart failure. And we also saw that mechanical unloading of the heart of patients with hypertrophic cardiomyopathy increases CTCF level. Okay, so once that we got our knockout animal, we started working in the epigenetic integrative analysis of heart failure. So when we started this project, what we did was to induce heart failure using performing TAC and also depleting CTCF. We isolate cardiomyocytes from the left ventricle of these hearts, and then we perform RNA-seq to study change in gene expression. We perform Hi-C to study change in chromatin structure, and bisulfite sequencing to study change in DNA methylation. So RNA-seq and bisulfite sequencing are two techniques that are very well known, so I'm not going to focus on them. But let me talk a bit about Hi-C because it's a bit more complex technique. Okay? So Hi-C is this technique that allows us to detect both intrachromosomal interactions that are taking place in the nucleus. Okay, so the first step in this protocol is to fix the DNA using formaldehyde. After that, you digest the entire genome using a restriction enzyme. In our case, we use EMBO1. And the next step is to fix the 5' overhangs using a clone of fragments and DCTPs that are marked with biotin 14. So after this step, we perform a ligation reaction that is going to generate circular molecule of DNA like this one that are conformed by the two regions that were interacting in 3D. Okay. After this ligation, we just sonicate the sample, perform a straight tapidine pull down to enrich our samples on these fragments that contain the two regions that are interacting in 3D. We prepare the libraries and perform the sequencing. So when we get the data from the sequencer and after the analysis, we can then generate an interaction frequency matrix like that, where the first thing that we do is to divide, artificially divide the genome in a small fragment called bins, okay? So the size of this bin is gonna depend on the depth of your sequencing. So the deeper is your sequencing, the smaller is going to be the size of the bin, and the higher is going to be the resolution of the experiment. Okay, so after dividing the genome in this bin, then the only thing we had to do is to count how many times each of these bins of the genome have been found interacting to the rest of with the rest of the bins of the genome. So in this case, for example, this bin number 2 has been interacting with bin number 1, 33 times. Number 2 with number 3, 45 times. Okay, so we do the same with all the bins, with the combination of all the bins. And then we're going to have these numbers. What happened that because it's very difficult to see differences with these numbers, using numbers, what we normally do is to color code these numbers. So we're going to have something like that, where the higher number of interactions correspond to a darker red color. Okay, so this one is the interaction frequency matrix that you're going to have when we focus in a very small locus of the genome. Okay, but if we zoom out now and we focus in a bigger region, we are going to have something like that, where you can start seeing the characteristic high C profiles, the pyramid-like high C profiles. Okay, so as you can see here, the top and the bottom part of this matrix are identical. So what we normally do is to remove this bottom part and display here the genes that are encoded in this region of the genome. So the first thing that you notice when you perform this high-C experiment is that the genome is being organized in units called TADs, topologically associating domains, where the interaction frequency within these TADs is much more higher than between TADs. Okay. So what does suggest? This one suggests then that in 3D, what is happening is that the genome is being organized in these glomerular structures that facilitate the interaction of the genes that are located within the TADs, but doesn't facilitate the interaction between genes that are located in different TADs. Okay. Another important feature that appears when you perform these experiments are these valleys between the TADs. These regions are called TAD boundaries. And its preservation is very important because if you disrupt them, you are going to induce a change in chromatin topology in this region that can affect gene expression and therefore the phenotype of the cell of the organism. Okay, so then when we perform high-C comparing our control, the CTCF knockout and the TAC, what we found is that heart failure induces a massive loss of interchromosomal interaction. It's something that you can see here in this heat map. Okay, so here I'm showing you the top 50 genes that are differentially expressed in both CTCF knockout and TAC animals. And then I sorted these genes by the number of interactions that are coming out from the gene. So you can see here that when we induce heart failure, no matter how, depleting CTCF or performing TAC, the number of interactions decreases in all these genes. Okay, showing that we have this massive loss of interactions. So this one here is the similar thing. It's a similar thing that what we have in the heat map, but focus in one gene. So in this case, we are focusing in the potassium channel KCND2. Okay, so here in the top part, you have the interaction frequency matrix for the locus where this gene is encoded, and then here we have the significant interaction, the fit high C significant interaction detected in this region. Okay, so each of these lines is representing the interaction between two regions. So this one, for example, is representing the interaction between this part of the genome and this other part. Okay, so as you can see here, when we induce heart failure by depleting CTCF or performing TAC, the number of interactions got lost. Okay, so you have a reduction in this number of interactions. And then this number of interactions, the reduction in this number of interactions correlates with change in gene expression. So in this case, this gene is being downregulated. So we saw that there is a very good correlation between loss of interaction and change in gene expression. But the direction of the change is not always the same. It's something that I'm showing you here. So you lose interactions, but it doesn't mean that the gene is going to be always downregulated. So in some cases, the gene is being downregulated, the red genes here. In some cases, the gene is being upregulated. So it's depicted here with the green color. So that's the case, for example, of MPPB gene. Okay. So here again, I'm showing you the interaction frequency matrix for the locus where the MPPB is encoded, the interaction, the significant interactions, and the gene expression. profile. You can see here how when we induce heart failure by depleting CTCF perform intact, we are losing interactions, and this loss of interaction correlate with an increase in the expression of the gene. Okay, so when we studied the enhancer promoter interaction, what we found is that this massive loss of interaction is also affecting this correlation, this feature. Okay, so this is something that you can see here in this heat map. So here I'm showing you again the top 50 enhancer promoter interaction, this time sorted by the number of interactions that are coming out from the enhancer and going to the gene. Okay, so here you can see again that when we induce heart failure, depleting CTCF of perform intact, the number of interaction is getting reduced. And again, this one correlates with a change in gene expression. So this one is an example of this. Okay, so here we are focusing in the gene ROCK2. So this gene ROCK2 have two enhancers, enhancer 1 and 2, that are interacting with the gene in the control, but this interaction are lost when we induce heart failure. Okay, and in this case, this loss of interaction correlate with downregulation of the gene. So, like I mentioned just before, there is a very good correlation between loss of interaction and change in expression, but in this case, again, the chain of this expression doesn't go in the same direction all the time. Some of the genes are being upregulated, some of the genes are being downregulated. The next thing that we did was to study the transcriptional behavior of the genes that are at the other side of all the interactions that are coming out from the differentially expressed genes that we found after inducing heart failure by depleting CTCF or perform intact. Okay? So this experiment, what this experiment saw is that upregulated genes after inducing heart failure tend to interact to upregulated genes, and downregulated genes after inducing heart failure tend to interact to downregulated genes. Okay? Suggesting that heart failure promotes a genome reorganization where genes with the same transcriptional behavior tend to get close to each other, tend to cluster close to each other, okay, after disease. So when we study distribution of the TADs after performing after inducing heart failure, we didn't see very big difference in the distribution of the TADs. But when we focus in boundary strength, we saw very big differences. So it's something that I'm showing you here. So here I'm showing you the location of all the TAD boundaries that were detected by our Hi-C data. Okay, so each of these lines is a TAD boundary. So if the line is blue, means that the strength of the boundary got reduced after heart failure, after inducing heart failure. If the line is depicted in red, means that the strength of the boundary increases after heart failure. Okay, so when we study the correlation between gene expression and boundary strength, what we found is that while the upregulated genes tend to be close to weak TADs, non-differentially expressed and downregulated genes tend to be close to strong TADs, okay? Suggesting that boundary strength influence gene expression. So you have a higher probability to find non-differentially expressed genes or downregulated genes close to stronger TADs than upregulated genes close to these strong TADs. Okay? Another thing that we can study when we perform Hi-C is the AB compartmentalization. So AB compartment are these regions that I mentioned at the beginning of my talk, where the genes that are located in these regions tend to be activated or transcriptionally active in A compartments or repressed in B compartments. Okay? So when you perform Hi-C, you can generate this AB compartmentalization profile and study the chain. That's what I'm showing you here. So here on the top part, we are, again, I'm showing you the contact matrix for this specific locus of the genome. Here you have the distribution of the TADs, where the strength of the TAD is depicted by the intensity of the green color. So if the green color is very intense, means that the strength of the boundary is high. Okay, and here you have the AB compartmentalization profile for control, CTCF knockout, and tag. And here I just so I'm showing you the genes that are encoded in this region of the genome. So you can see here, when we induce heart failure by depleting CTCF of performing TAC, there are some regions, like these ones here, that in the control they were in an A compartment, and now they move to a B compartment. Okay, so this one is going to have an effect in the transcriptional behavior of the genes that are located in these regions. So when we study AB compartmentalization in a global manner, what we found is that this feature is very stable. Okay, so what we found is that only 5% of the genome was able to move from one compartment to another. But the correlation between AB compartmentalization and gene expression was very good. Okay, so here, that's what I'm showing you here. So here I'm showing you how, after depleting CTCF, after inducing heart failure by depleting CTCF of performing TAC, the genes that end up in an A compartment tend to be upregulated, and the genes that end up in a B compartment tend to be downregulated. Okay. Another thing that we are doing right now is trying to correlate DNA methylation with the AB compartmentalization. So the idea behind this experiment is to try to correlate A compartment with hypomethylation and gene expression, and correlate B compartments with hypermethylation and gene repression. So we are still working on this project, but what can I tell you right now is that that 99% of the CPGs that were differentially methylated in both after both CTCF depletion and TAC are changing in the same direction. What does it mean? That after inducing knockout or TAC, the CPGs that are changing in these two states are following the same trend. In both cases are hypermethylated, or in both cases are hypermethylated. So there is only a very small percent, only 0.9%, that don't follow this trend. Okay, so 59% out of this 99% were found hypermethylated. Okay, suggesting that heart failure induced hypermethylation, at least in this set of CPGs. So when we study the correlation between CTCF, between DNA methylation and AB compartmentalization, what we found is that after inducing heart failure, all the compartments, okay, the percent of methylation in all the compartments after inducing heart failure, no matter how, performing TAC or depleting CTCF, is higher than in the control. Okay, and then if you compare B compartment with A compartment, what you see is that the percent of methylation is always higher in B compartments than in A compartments. So we continue working in this experiment, try to confirm these results. So hopefully we will be able to give you more data in the future. So finally, another thing that I would like to mention is that all this high-C data that we generate with our data can be used to generate probabilistic 3D models like this one, where we can represent where each of these spheres represent one of the TATs that conform the different chromosomes. Okay, so here I'm showing you the entire genome, and here I'm showing you only chromosome 1 and 2. So each of these spheres here represent one of the TATs that conform the chromosome 1, and these ones are the TATs that conform the chromosome 2. So this model have proved themselves very useful to analyze different chromatin structural features like the AB compartmentalization, for example. So using this model, we have been able to study differences in nuclear organization between organs. So in this experiment, for example, what we did was to generate the 3D models using high-C data from liver and the heart, and then study the probability of a TAT being towards the interior of the nucleus versus the periphery. Okay, so to do that, what we did was to divide the nucleus in 5 concentric cells, and then study the probability that you have to find an A TAT close to an A TAT in each of these regions. Okay, so let me tell you. So in the heart, then what we found is that the probability that you have to find an A compartment, an A TAT close to an A TAT, is high in the center of the nucleus, and this probability decreases as we move to the periphery. For the B TATs, the inactive TATs, we saw the opposite trend. So the probability to find 2 of these TATs close to each other is higher in the periphery, and decreases as we go through the center of the nucleus. Okay? So this one is the expected trend, because as everybody knows, the periphery of the nucleus correlates with gene repression, and the central part of the nucleus is correlated to gene transcription. Okay? But in the liver, we saw the opposite trend. In the liver, what we found is that the probability that you have to find two ATADs close to each other increases as we go to the periphery, and the probability you have to find two inactive TADs close to each other increases as you go to the center of the nucleus. Okay. So right now we continue working on this thing, but basically what this experiment are showing is that different organs are having different nuclear organization. So as a summary of the second part, we can say then that heart failure induces massive loss of interchromosomal interaction, including enhancer promoters interactions. We can also say that changes in gene expression detected during heart failure are related to alteration of TAD boundaries. I also show you that heart failure induces gene reorganization of differentially expressed genes. Also that heart failure alters AV compartmentalization. And finally, we also saw that AV compartmentalization correlates with gene expression and DNA methylation. Finally, I would like to thank everybody in the Vondriska lab, especially Douglas and Tom, because they're always there helping me with the experiment and the grants. And of course, I would like to thank all our collaborators, Dr. Niels Galhard, that is working at Erasmus MC in Rotterdam, Dr. Jibin Wong and Frank Albert, that are working here with us at UCLA, Dr. Enrique Valderas, that is working in the University of Utah, and Dr. Vin Ren, working here in UCSD in San Diego. So, yeah, that's it. Thank you, guys.
Thanks you very much, Manuel. Thanks for your great talk. So while people are writing in with questions, which they have been gradually, maybe I can start off with a question. I just want to make sure no one's going to ask it already. Yeah. I was wondering, I don't remember how quickly or if you did the knockout of CTCF in relation to the TAC, but how quickly do these changes occur? Do you know if CTCF induced very quickly with hypertrophic stimulation in hours, minutes, or is it more something that's associated with really pathology kicking in as opposed to the adaptive phase of hypertrophy?
Yeah, so in our case, for example, our model, we get the 80% depletion after 5 weeks tamoxifen treatment, you know. So all these changes that we are seeing here are 5 weeks, all changes. But there are other people that have done similar experiment in cell lines. Benoit Bruno, for example, they also knock out CTCF using the degradome system and a cell line, you know. So what these guys saw is that when they activate the degradome by adding the auxin and they deplete CTCF, these chromatin, these chromosomal changes are very fast, you know. Mm-hmm. As soon as you remove CTCF, you start having changes in chromatin structure. So the nice thing that they have is that they are able to achieve 100% depletion of CTCF. So what they saw is that when you completely remove CTCF, the TADs totally disappear. And then when you recover CTCF level by removing this auxin, you know, you get the TADs back. Okay. So it should be a pretty fast process, you know, but we don't catch that because our system is 5 weeks.
Okay, I can see it's hard to tell. Okay, maybe we can start with some questions then. So the first one from an anonymous attendee asks, how long does it take for CTCF conditional knockout hearts to develop cardiac dysfunction? Yeah,
so, okay, so we start seeing problems in the heart after 3 weeks, you know, and the hearts became hypertrophic after 5 weeks. The thing is that if we wait longer than 5 weeks, the mice start dying. So the survival rate drops. So we don't have enough mice to do experiments. So that's why even when we are not able to deplete 100% of the CTCF in these animals, we had to do the experiments after 5 weeks. Yeah.
And they don't then start dilating, they just stay in hypertrophy and then die?
Yes, correct. So yeah, the heart of the animal is huge, you know, and it's barely beating, but when you look at the animal is very happy. But if you wait 1 more week, all of them die. So.
Thank you. So the next question from another anonymous attendee asks, did you by any chance check if some DNA loci are moved towards a nuclear pore complex, either in control or diseased animal?
No, we didn't do that, but you know, there's a thing that we would like to do. Yeah, so correlate change in chromatin structure with lamina associating domains, for example. So what we did in the lab was to check the opposite thing, you know, how genes that are being activated move toward the center of the nucleus, but we never check in the other direction. But yeah, that's a thing that we could do in the future.
Okay, thank you. The next question from Johannes Silva, like somebody you know, says, hi Manuel, great talk. Did you test the effect of CTCF overexpression in heart subject to TAC? Maybe an overexpression would lead to a better outcome.
Yeah, so that's exactly an experiment that we're doing right now, you know. And what can I tell you is yes. So right now, what we have seen, preliminary data, is that when you overexpress CTCF in the heart, the TAC phenotype is not as strong as when you have less CTCF. Yeah. But we're still working on it.
Okay, thank you. The next question from an anonymous attendee again is, for A to B transition in CTCF knockout versus control, did you see this change in any particular set of genes related to the heart? Did you do any gene ontology analysis on those changes that on those genes that show changes in gene expression?
Yes, we did gene ontology on these genes, and there are tons of genes that are related with cardiac terms. Yeah. So that's another thing that we are doing now, is going to this, you know, because we did this massive experiment, and now we have a list with tons of genes. So now the thing is that it's very difficult to go and focus on only one of them, you know. So now what we're doing is going to, after the more promising one, and try to go deeper, you know, with them. But yeah, we have a lot of cardiac related genes.
Okay, thank you. The next question from Gabriella Schiattarella, hope I pronounced that correctly, Gabriella, is, it looks as though TAD movements, allow me the term, in the nucleus are more extreme in the heart than in the liver. What's the reason for this? Yeah,
we don't know yet. We really don't know yet. We are working on that. We are trying to improve the resolution of our models, you know, to try to figure out what is the difference between the tissues. And we are also trying to add new tissues to compare with liver and the heart. We are working with the brain and try to see if we can find some kind of correlation between the three of them, you know, but right now we don't know.
Could be something to do with the regeneration.
Yeah, probably, yeah. It would be lovely.
The next question from Sina Hadipour Lakamisari asks, thank you for your great talk. I have a few questions. So maybe I'll start with the first question. Do the CTCF knockout mouse have any other health problems? And have you tried doing TAC on the CTCF knockout mice?
Yes, we did TAC on the knockout mouse, and the mice die. I mean, they don't, it's completely, I mean, we don't have any die survive, you know, after performing TAC on these animals. But other than that, we didn't see any other problem, you know. We are also studying now the metabolism of these animals to see if we can see something, but we didn't see any different right now. So no, there's nothing else.
Okay.
Sorry, just to check. When you say you're studying metabolism, what are you doing? What kind of experiments are you doing?
Well, yes, we are doing glucose intake, you know, insulin. Right now, that's the two things that we are doing right now. And then if we get something there, we will try to do other experiments, you know, but basically that's the first thing that we are checking. We are interested in that too, because when we check the RNA-seq, there is a lot of genes that are involved in metabolism that are changing in gene expression, you know. So that's why we would like to go in this direction too. But,
thank you.
Thank you. So the next part of the same question asks, do you have any CTCF mutations reported in heart failure patients?
Yeah. I know that there is CTCF mutation in tumor, you know, in prostate tumors, breast tumors, in wheel tumors. You have mutations so the thing is that when you have mutation in the same finger domain of the protein, you have these tumors, okay? If you mutate the binding site for CTCF, mutation in binding site of CTCF have been found in some leukemias, okay? And if you have upload deficiency of the CTCF gene, you have brain problems, But in the heart, we didn't see any mutation. We also checked GWAS, and we didn't find any GWAS that correlates CTCF with heart failure with any variant. So
okay. Yeah. And a third part of the same question, or the same question is, have you looked at the pattern of CTCF expression over time with the normal TAC model, and does it gradually decrease, or is there a sudden drop after a certain ejection fraction threshold, for instance?
So that's a good idea. That's a good question. I've never done it, and I have the data. I have the data, but I never compared them. I don't know. I really didn't think about this thing, but I could do it. Yeah, that's not a problem. Just to see if the CTCF depletion is inducing heart failure faster than TAC. Yeah, that's a good idea. I can do it. Yeah, thank you.
Okay, thank you. The next question from Jessica Flegger asks, with an overall reduction in CTCF during hypertrophy and failure, what molecules do you think might be responsible for the regulated clustering of up or down regulated genes in clusters or compartments? Does that make sense to you?
Yeah, so this one, to do that, what we have done Homer analysis, to see what genes are being binding the promoter of these clusters of genes, And right now, the thing is that we don't have very clear results. So that's why we didn't go in this direction. We have done the thing, but there is nothing there that caught our attention. So we prefer to focus in other directions.
Okay, thank you. The next question from Gidros Kanapouris, again, sorry for pronunciation, says, great talk. What is the potential mechanism by which heart failure leads to chromatin reorganization? CTCF.
Hopefully CTCF, we don't know yet. So that's the thing that we are trying to figure out, So hopefully CTCF, So that's our hypothesis. Our hypothesis is that alteration of CTCF level is behind of all this chromatin structure, changing chromatin structure during heart failure.
Okay, thank you. The next question from Anna Gans says, very clear talk. How about how does A to B compartmentalization relate to euchromatin, heterochromatin?
Yeah, so here we have a very good correlation, yes. So when we have changed this thing, there is a very good correlation between A compartment and active histone marks, and B compartment with repressive histone marks. So yeah, this one, in our data, at least, there is a very good correlation between that.
Thank you. The next question from Viba Rani says, nice work. Just for curiosity, microRNAs play critical roles as epigenetic regulators. Did you establish any microRNA CTCF associations in hypertrophy? There's several parts of the question. Maybe that's the first start.
So the answer is no, we didn't do that. But when we performed the RNA-seq, yes, there was a lot of microRNAs that are which expression is being altered after depleting CTCF for performing TAC. But we didn't go up to that.
Yeah, so I guess that kind of answers the next part, which is they've asked, does CTCF regulate non-coding RNAs? That in breast cancer cells, CTCF is known to regulate some microRNAs. So I guess you pretty much answered that question. So you've definitely seen some, but you haven't yet looked at them.
Exactly. But we didn't do the experiment.
Okay, thank you. The next question from Antoine Mouchier asks, when you think of inherited cardiac disease like titin truncating mutations or phospholamban mutations, would you expect that gene therapy restoring cardiac function, I guess by overexpressing CTCF, would also impact the genome reorganization?
So that's the hope. That's the hope, So that's why we are running these experiments right now, where we are overexpressing CTCF to see if they are able to avoid the effect of TAC, And another thing that we are doing is putting back CTCF to see if we are able to recover the original chromatin structure that we have in the heart. So we are going in this direction right now. Let's see what happens in the future.
Can I just ask both of you actually in this area? So has anybody done an experiment whereby they've treated the mice with beta blockers that have been under TAC, and then looked at whether the treatment reverses some of the epigenetic or chromatin changes? Has anybody done that, or?
I'm not sure with the standard medication that's on the market now. some maybe not pharmacological treatments, but some manipulations that can reverse the phenotype, they might reverse gene expression changes. They don't always reverse all the epigenetic changes.
Okay.
There's sort of an imprint.
Or the genome reorganization, sorry, for example. I mean, it's related to this question here in a way. You know, because that's your hope.
As far as I know, nobody had done this experiment. Yeah.
I'm just wondering what the plasticity is of that genome reorganization that you might see. Is it, you know, can it go in both directions?
Yeah, exactly. So you have a change in gene expression with the disease, and then when you treat it, you come back to the original state. Yeah, this one is beautiful. You know, that's the kind of experiment that we're trying to do now with CTCF, overexpressing CTCF. You know, but right now we don't have still data.
Okay, thank you. The next question from Heinrich Teigmeier asks, can you speculate whether exposing the heart to a specific defined metabolic environment can alter chromatin structure and function? And if so, how?
I don't know. I mean, I really don't know. But I guess so, yeah. I mean, again, as far as I know, nobody had done this kind of experiments, but why not, you know? I mean, if metabolism is able to alter genes that are involved in chromatin structure in some way, yeah, you're going to have changes, you know.
Have you looked at any metabolic syndrome models of CTCF expression?
Not that I know, yeah. There is no correlation between CTCF and metabolic problems that I know, that I'm aware, you know, but I don't know.
Okay, thank you. Next question from Emma Monte asks, does losing enhancer promoter interaction as in heart failure affect the energy required or efficiency to transcribe affected genes?
So you're asking if the loss of interaction is altering gene expression?
Ask the energy required to transcribe the genes, maybe because the conformation is not conducive to gene expression.
So what we saw is that, yes, when you lose interaction, when you are losing the interaction, the loops between the enhancer and the promoter are lost, and this won't have an effect in gene expression. What we have seen also is that you have loops that are, you have conformational loops after inducing heart failure, you know? So there is a chain in chromatin looping that is altering gene expression, but I don't know if that's what Emma, another question.
Next question. Yes, Rose Yin asks, says, thank you for your lovely talk. I know that TAC surgery results in heart failure in male rats, but has relatively little effect in male rats, but relatively little effect in female rats. Is CTCF knockout model equally viable in male and female? You use mice, didn't you? Yeah, but.
Yes. So when we started this experiment, we did a pilot experiment comparing female and males, and we didn't see any different. So we decided to go after the big experiment combining both of them. You know. So yes, there is no our experiment didn't show any different.
Okay, great. Thank you. The next question from Marta Sulik. Says, again, fantastic talk. I have a few questions, if that's okay. Number one, what is, we sort of answer this a bit. What is the timeline of chromatin architecture changes in TAC? And have you looked at different timelines? And would you expect differences or similarities in chromatin structure in failing hearts that was induced by TAC or myocardial infarction? Sorry, that's the second part of the question.
So I would love to do this experiment. So timelines to see how chromatin structure start changing, you know, with the disease. The problem is that these experiments are super expensive, and you know, HICE is very expensive technique. So what we did is to go with the time points that we know that we're going to have a difference, you know. But yeah, I would love to do this experiment, and I'm sure that it's not a 0%, 100% change. You know, it's going to be a gradual change in chromatin structure. But yeah, we didn't do this experiment.
Well, we can certainly look at how when CTCF starts to go down anyway, and how soon.
Yeah, so that's what Benoit Bruno paper is showing, you know. So as you start losing CTCF levels, you can start see how the TACs are fading, you know, and disappearing. So yeah, this data support this theory.
And have you looked at a myocardial infarction model?
No.
No. That's a good question. Does the chromatin architecture change the same in all heart, cell types of the heart, or all cells of the heart?
That's another thing that we are doing right now. So we are trying to do Hi-C in different population of the heart, and then try to see if there are differences there, and also generate 3D models to see if chromatin structure changes among the different cells that are part of the heart. So we don't know yet.
Single cell Hi-C is still, but
I know. Yeah, so the thing is that I'm not expecting too much difference, because that distribution is a very stable feature, So when you compare different cell lines, the TATs the TAT profile is almost the same in all of them, So I don't expect too much dramatic changes, But let's see. Especially when we will induce heart failure, I don't know, maybe there we will be able to see bigger changes.
Okay. Maybe between different chambers of the heart, you may expect more. I don't know, because of the different stress.
Yeah, so that's another experiment that is in the line. It's like compare right ventricle with left ventricle, for example. Yeah, this one would be very nice to do.
Okay, thank you. The next question from Ursula Ravens says, "Thank you for your very clear talk. How did you also find changes in genes that might regulate arrhythmias at all, or atrial fibrillation involved?"
So we have, yes, when we perform the gene ontology, there is a couple of terms that are related to arrhythmias, yes, and we have some genes that are related to that. But again, we didn't do anything with that yet. Yeah. The thing is that this kind of experiment, release so much information that, then it's impossible to go after all of it, So we are also looking for collaborators, to try to, provide this data, and then maybe they can do something that is related to their project. So if somebody is interested in collaborating with us, just please contact us, and we will try to work together.
But again, sorry, as a naive sort of, person in this field, and this is maybe to both of you, to what extent can you extrapolate the information from the left ventricle with sort of a disease of essentially left atrium? Can you do that? And would you be comfortable doing that?
As we've also got sequencing from different chambers and seen definitely do differences, and as well in different etiologies of heart failure.
Sure, but in this case, you obviously just taking left ventricular cardiac myocytes, and that's it, right?
Yeah, so in this case, we don't do it, So we wouldn't do that. I don't feel comfortable doing that. So I would prefer to do the experiment that you are saying when we do Hi-C in different regions of the heart. So that's why we always mention left ventricle, left ventricle, left ventricle, to make it clear.
Were the atria dilated at all in the CTCF knockout?
In some of them they are, yeah. Not in all of them, but in some of them, yeah.
Okay, thank you. We'll move on to the next question. From Alessandro Botero, says, "Great talk, Manuel. Have you seen differences in intrachromosomal interactions in TAC CTCF knockout mouse, and just the percentage of the contacts change?"
So that's a very good question. The thing is that the problem with the Hi-C is that the interchromosomal interaction, we are working with the interchromosomal interactions. The problem is that the Hi-C is a technique that provide interchromosomal interactions that are not very good, because the background is so high that it's very difficult to get only the significant ones. So when we work with the significant ones, we don't have too many of them. So maybe the data are not going to be very reliable. So that's why we mainly focus on intrachromosomal interactions. But yes, we have data with the interchromosomal interaction. The only problem is that we don't feel comfortable yet releasing this data because of the way how we analyze the data, But I would love to collaborate with somebody that can help us with that. It's a big thing.
Okay, thank you. The next question from Llewellyn Roderick says, "Great talk. Would you expect the chromatin organization and TAD boundaries are altered in laminopathies or progeric diseases?"
Yeah, so it's well known that lamina-associated domains are related to TADs, That's another thing that I would love to do. And in fact, there is a person now in the lab that is trying to study this kind of correlation, change in TADs or chromatin structure with the lamina-associating domains. But right now, we didn't do anything. Yeah. So what I can tell you is that the lamina-associating domains are attached to the nuclear lamina thanks to CTCF. So CTCF is attaching this lamina-associating domain to the nuclear lamina. So when you remove CTCF, the lamina-associating domain is moving towards the center of the nucleus. So you are expressing new genes, and this one is generating diseases sometimes. Like I think that they do laminopathies. There is a laminopathy that is that correlates with muscle problems, muscle dystrophy. Yeah. So muscle dystrophy because of that. Yeah. Mutations of the region that attach the lamina-associating domain to the nuclear lamina.
Yeah. Okay, great. The next question is from an anonymous attendee, but they say, thank you for the talk, Manuel. And a simple question, did you do the computational analysis yourself or did you collaborate with the bioinformatics?
Yeah, so with this one, I'm very lucky that I have Douglas Chapsky in the lab, that is the person that is working with me in all this analysis, So basically what we do is we ask a question, and then he tried to figure out how to do it. We are fighting all the time, until the end we get the data that we like. Then we talk to other people, we collaborate with other people. We have collaborated with Dr. Bin Ren, for example, in San Diego. They are expert in high C. So they help us a lot, But basically, Douglas in the lab is the person that is taking the lead in all this analysis.
Okay, yeah, thank you. The next question from Antoine Mouchier asks, does chromatin reorganization in heart failure hamper gene therapy efficiency? So is it like a rate limiting, or not rate limiting, but something you have to...
We don't know yet. That's a thing that I would love to do in the future. Try to figure out system to modify chromatin structure because the thing is that right now we have mapped all the changes in chromatin structure, so we know the regions that are being modified. So the next step now will be to develop tools that allow us to go to these specific regions and remodify them to make them come back to the original state. Using, for example, Cas9 system, So let's see in the future, yeah. I don't know. There is some Cas9 system that help you to methylate regions that are unmethylated, for example, There have been developed some techniques now that allow you to generate loops or disrupt loops. So the future looks nice. So let's see.
The specificity is always a problem with these.
Yeah, exactly. That's the main problem right now.
Yeah. Okay. Emmanuel Tampakakis asks, have you overexpressed CTCF in any heart failure mouse models to potentially rescue the phenotype? I think we've covered that. And do you have more mechanistic data on specific heart failure genes that mediate CTCF in the heart?
Yes, we have a list of genes which promoter is bound by CTCF, and then we know what genes are losing the CTCF binding after heart failure. So we have the list there, and there is very interesting genes related with cardiac disease. But again, we didn't do anything yet with all this data.
Yeah. What about genes that actually regulate the CTCF itself, the gene?
So the nice thing is that CTCF auto-regulates itself. It's regulates itself, So, when you have less CTCF activate the transcription of CTCF. The thing is that because we deplete the entire gene, there's no way for the gene to auto activate the transcription.
I was wondering that. Okay. Oh no, we have another question.
There are also, Emma, there's a couple more in the chat box.
Earlier, okay. So I'll just ask this one and then maybe we'll move over to the chat box.
Yep.
Any data or plans to do chromatin 3D capture on human heart failure samples?
Yeah, so this one, I know that Roger Fu is working on that, so right now we don't want to enter in this, we don't want to take this direction, but yes, I think that I would love to do in the future, even collaborating with them or something. Yeah, the thing is that here at UCLA, we have very nice systems, so we can have samples, so we could run all these experiments. Yeah, but right now we are not going in this direction because we know that Roger is working on that. So.
Okay. Let's move over to the few in the chat box. Mickey Alexanian asks, that's great talk. What do we know about, what regulates CTCF in heart failure? We'll just, auto-regulates. and you show depletion of CTCF in many strains, which suggests that CTCF is highly regulated.
Yes, that's correct. So yeah, that's correct. So the nice thing that we saw in this experiment is that there are some mouse strains where you don't see a depletion of CTCF, a reduction of CTCF level. What you have is an increase, you know. So that's another thing that I would like to do, is to study the differences between these mouse strains. Why in these strains you have, after inducing heart failure, you have more CTCF, you know. These strains are weird, you know, compared to the other ones. But that's, yeah, there's a lot of things to do.
Are they ones commonly used in research or not really?
Some of them they are, yeah. Some of them they are, yeah.
Okay, another question, I think a few of them have already been transferred over to the main questions, but one of them, which I don't think we've covered, from an Enrique I can't see the full name, Valderas, asks, could you elaborate on the potassium channel that you found regulated in your model? What would be the role for this channel in heart failure or chromatin remodeling?
Yeah, so I cannot elaborate because I didn't check anything, but Enrique, if you are interested, let me know, because we have a list of channels which expression is being altered, you know, after CTCF depletion. I told you a lot of times, we can do the experiment whenever you want, you know, collaborating with you, that you are the expert. We didn't do anything. Yeah.
Okay, excellent. I think we've come to the end of all the questions. I'm just having another quick check that we haven't missed anything. And excellent, perfect timing.
1 hour, yeah, perfect.
Yes, so thank you very much, everybody, for watching, all your questions, and thank you to Manuel. Great talk. And
thank you, guys.
Thank you.
And for reading all the questions. You know, it's very nice when people read the questions. Thank you.
And we'll see you tomorrow.
Well, okay. On my behalf as well, thank you both. Manuel, I've learned really a lot today, and Emma, thank you for amazing sharing. Your knowledge and help was really greatly needed here, because I know nothing about this. So thank you, everyone, and see you all tomorrow, hopefully.
Thank you very much. Bye.
Bye.